So Robert's going to talk to us today about 13 years of cover crops and rotation history. What does this do for our soil health? So Robert is a research fellow at the University of Tasmania, and he works within the Tasmanian Institute of Ag, and his work focuses on plant pathological problems in vegetable crops with a strong emphasis on potatoes. He also investigates soil-related issues that impact productivity and crop health. Thanks, Robert.
Okay, thanks for that introduction, Sue. So today's topic is entitled 13 Years of Continuous Cover Cropping. What does this do for our soil health? We've got a team from TR that are working on the project. There's myself. We've got a microbial ecologist, Shane Powell, some soil scientists, John McPhee, Richard Doyle. We've got a PhD student, Bernard, and Callum, who you heard from earlier, and also Phil, who does a lot of the work on the site. The trial's been managed over a 15-year period, and it's also included farm managers that have looked after the site. So we have the person that initiated the site and looked after it was Lyndon Butler, who's here today. Then we've had Vaughan, and in recent years we've had Doug Clark managing the farm and managing the site. For a 15-year trial, we've needed to utilise various funding sources. So the work was initiated by Dr. Lee Sparrow in 2005-06 when he received some funding through Hort Innovation. And this was primarily to look at potato pathogens in the site and track them over time. So that received funding for 5 years. Then he received some more sporadic funding from 2010 to 2014. Then essentially the site lost funding, but from 2015 to 2016, Simplot through Frank Mulcahy were kind enough to initiate and maintain some pathogen tests on the site, which we're highly appreciative of. In more recent times, the project is funded by Aggregate Tasmania, and this has enabled a range of scientific measurements to occur, particularly over the last 3 to 4 years. We also have a small fund from Hort Innovations that's supporting a PhD project in the area. So the history of the site was chosen 15 years ago. So there's an overview of what Forthside looks like. Essentially, we have 6 major paddocks at Forthside. The one we chose is in the far corner. It's not the best site, it's probably the hardest to crop and manage on the farm. It's got slope and aspect variation across it. Does have the same soil type, being a ferrosol. Approximately 5 hectares in size, and highly suitable for long-term trial work. So that's why it was chosen by Lee Sparrow 15 years ago. Essentially, that's an aerial overview of it. It's been set up into 4 blocks, 4 replications. So on the southern side, we have Rep 1 and Rep 2. On the northern side, we have Rep 3 and Rep 4. In terms of site variation, there is a little bit of site variation across the site. This black line down the middle there is potentially where there used to be a fence line or a different practice maybe 20 to 25 years ago, because we have observed that the pH on the left-hand side is 5.9 and 6.5 on the right-hand side, so there may have been some lime added to different aspects of the paddock maybe 20 years ago or so. The setup is pretty simple. It's basically comparing 3 winter treatments. So one of them is a biofumigant, called Caliante, the other is a ryegrass, and the third is a fallow operation, which is leaving it as bare ground. And these are fairly common ways of managing soil over winter within Tasmania. And there's 4 large plots of each treatment that we can see there on the diagram. So our cover crop treatments, just a couple of points about them. They're both monocultures, so our Brassica caliante is sown as a monoculture. Our ryegrass is also sown as a monoculture. For those that don't know much about cover crops, they're a non-cash crop and grown as an alternative to a fallow. And the biofumigant has extra properties. It produces compounds such as glucosinolates in the leaf tissue. These then get incorporated into the soil and release a biofumigant called isothiocyanate, and this has the potential to target pests, pathogens, weed seeds within the soil. So essentially, this rotation has been occurring for the last 14 years. So we have the same winter treatment of either fallow, green manure, biofumigant, but we do have a spring summer crop, we go back, that is sown over the whole site. And that's generally what fits our commercial rotation. It may be a carrot, potato, a broccoli. This year it's beans. That's just a pictorial of the setup. The plot size is quite large, 80 by 36 metres for each individual plot. These can be broken up into subplots. So overall there's 48 experimental units across the site. We've focused on measuring physical, chemical and biological soil properties, specifically over the last 3 or 4 years. Most of our soil sampling is at a 0 to 15 centimetre depth. Some of the types of things that we've measured key soil pathogen data, particularly DNA data related to predictor tests. So that's mainly looking at potato and vegetable-based pathogens. A newer tool we've used over the last 3 to 4 years is soil community analysis. We've looked over most years at soil chemistry with a major focus on soil carbon. We've also looked at specific plant chemistry to make sure that our biofumigant Caliante actually produces glucosinolates. Physical parameters, we've looked at structure in terms of using wet sieving techniques. We've also looked at compaction patterns by measuring with penetrometers. We've also looked what happens in terms of weed specifics at the sites, in terms of densities and seed banks. And I guess the main thing for most farmers is that we've looked at what happens from a commercial perspective in the following crop in terms of yield and disease outcomes. So I'm going to present just a snapshot of some of these results today. So first of all, for chemistry, this is a typical type of chemical profile that we get when we do a soil test. Most farmers get soil tests done for NPK and they can get recommendations of various fertilisers from that. I really only want to focus on one point in this, and that's carbon. So carbon is a good measure or surrogate of soil health. So in 2005-06, the carbon levels across the site were approximately 4.2 to 4.3% across the fallow, the biofumigant and the ryegrass. Over that long-term rotation, they've gone up to 2012, they've gone up a little bit. The last years where we've had more extensive measurement, they've tended to trend down. The main point to observe is that initially all of them started off at a similar level, but as we trend through, the fallow is always at a lower level. That's what you'd expect. It's generally at about a level of 0.2 to 0.5% less than the ryegrass or the Caliante. One of the reasons for that is, of course, we're adding organic matter to the soil. So this is how much organic matter we're adding to the soil. So in 2018, for the ryegrass plots, we are averaging approximately 8.5 tonnes per hectare, and Caliente 12.6 tonnes per hectare. So obviously that's going to provide some benefit to carbon levels when we're incorporating that into the soil. More specific data over the last 4 years, so that's our organic carbon levels there. We've also got organic matter levels. That's really just a surrogate for organic carbon. It's about 1.8 times what organic carbon is, so we've got a similar result there. Labile carbon, which is a very active form of carbon, is also higher in the Caliente and ryegrass treatments. Caliente is significantly higher than the fallow treatment. So it's indicating we're getting increases in carbon when we're adding organic matter, and that's probably an expected response, but it's good that we're actually measuring it and seeing how it changes with time. Some other things we're measuring across the site, we're measuring physical soil health. So we're using infiltration tests, you can see on the right-hand side there, penetrometer tests, where we're looking at soil penetration resistance. And these are the types of graphs that we get here on the left-hand side. I'm just going to have a look at a couple of results here, showing the results from our penetrometer work. So it's just two contrasting results, and it's showing the left-hand side in the topsoil that we're getting better penetration through the fallow. That was made in spring 2017. In winter 2019, through the whole profile, all the treatments showed a similar pattern in terms of penetration. So the penetrometer results haven't really shown us anything at this point in time. We've got another 8 results that aren't presented here today, but they've kind of been not showing a lot of conclusive results. Where we are getting results is in the aggregate stability tests. So an aggregate stability is really telling us how strong our soil is, how susceptible it is to erosion, and how it can cope with an external force. So it can be measured by many different ways, and typically a lab test might be a rainfall simulation test. We use a wet sieving process, which is on the next slide. We exert a force over the soil, and the soil breaks down into different categories. So we can just possibly play that video there, which is showing the wet sieving process. So it's a force exerted over soil. Soil breaks down into 3 categories. We have 2 sieves. One is more coarse on the top, then going down into a finer sieve, and then the rest that comes out the bottom is less than 0.25 millimetres in size, and that would be regarded as an unstable soil. So what we're wanting in a good stable soil is a higher proportion of soil particles greater than 2 millimetres in size. So just a snapshot of what we've got from one result. So we've got 7 or 8 results on this, maybe a little bit hard to see down the back, but essentially where we've utilised a cover crop, either grass or Caliente, we've got approximately 30% of our aggregates are sitting in the stable class, and within the fallow there's less than 20% of our aggregates are sitting within that stable class. We have a higher proportion of unstable aggregates coming out of the fallow. So that's indicating that we've got an improvement in soil structure, and that's a response of utilising cover crops. Just a quick study as to how structure can change quickly over time. It takes a while to build soil structure, but it can be degraded quite quickly. So this year at Forthside we had a carrot crop that went in, and it was harvested a little bit late. The soils were a little bit wet, and it resulted in a fair bit of damage to the soil. So there you can see some of the images there. A fair bit of crusting, compaction, and soil damage. So we've just recently got the results back from what that did to our soil. So pre-carrots, we had aggregate stabilities, stable aggregates greater than 2 millimetre of about 40%. Post-carrot, there's only 25% sitting in that class. So there's been a significant decline in physical stability of the soil through utilising a carrot crop that was harvested when the soil was perhaps too wet. This is just an overview of what some of the benefits of adding carbon and cover crops to the soil does. So the diagram on the right-hand side shows what happens after a rain event. So it shows what typically happens on the northwest coast. We have a lot of red soil that can end up on our roadways, end up in our waterways going out to Bass Strait. Cover crops are a process of reducing these impacts. Perhaps the novel aspect of this project is looking at some of the biology and how that changes within the soil under the 3 different treatments. So we can look at biology from a number of perspectives. We can look at it from a community perspective when we look at what communities are there, but we can also look from a pathogen or single species perspective. So in terms of a single species perspective, we're focused on some of the pathogens. So it may be a little bit hard to see from the back, but for Spongospora, we have levels in the fallow, 57 picograms, 85 under biofumigant, 120 under ryegrass. They're not significantly different. Just to testing, so we did look under the fence line and levels were pretty much near zero, and that's associated with the fact that haven't really got a crop growing under the fence line. Other pathogens that cause club root, so Plasmodium ophora brassica. Caliante is a host for that, so populations of that pathogen increase under Caliante. Just a real quick summary of some of the other pathogens. Rhizoctonia AG 2.1, which is a root nipping pathogen. Very small changes in levels under different treatments. Rhizoctonia AG3, which causes black scurf on potatoes. No significant changes across the treatments. Powdery scab was a little bit high under ryegrass, but not significantly different. Club root, substantially higher under the Caliante treatment. And Sclerotinia, little bit higher under ryegrass and Caliante. We should mention that Forthside is a pretty low to moderate disease site, so it hasn't got specifically high pathogen levels. And if we want to see some of the benefits from Caliante, some of the literature indicates that a lot of the benefits come in other species, particularly nematodes and that type of thing. So we don't have a lot of nematodes at Forthside. Just a quick case study of tracking pathogen levels and how they change over time. So an interesting thing was that we grow potato on a 5 to 6 year rotation here at Forthside. But in 2009-10, an extra rotation went in as part of Lee's work. So that's 9 years ago. So there you can see the plots up the top. We've got an onion crop at that point in time, but specific plots have been planted to an extra potato crop that we can see up there. And we wanted to see how that impacted specifically potato pathogen levels. So if we focus on the pathogen that Callum talked about in our last talk, Spongospora subterranea that causes powdery scab, we can see that pathogen levels track along and they rise up after a potato crop. So here, after that potato crop was grown, we have a spike in Spongospora. The crop that didn't have a potato crop has levels that are lowering down until the next potato crop was planted in 2012-13. So by the time we came to plant in 2012-13, this field here that had the extra potato in it was a lot higher risk for disease. After the potato crop, level spikes up again. 6 years later, it spikes up again. Basically, we've still got a residual effect of that extra potato crop coming to a higher level from 9 years ago. And that could be partly responsible due to the increase in volunteers that occur when we have a 3-year rotation. So potato volunteers are a problem on the northwest coast, and they maintain inoculum levels, and they are a lot higher levels from the 3-year rotation compared to the 5 to 6 year rotation. So that's a little bit about pathogen levels. If we go back and look at some more broader measures of biodiversity and soil community analysis, some of our studies conducted by Shane Powell has focused on 3 different community groups. This is known as soil community analysis. They've broken it up into 3 sectors: bacteria, fungi and eukaryotes. And this is looking more at a family level, so it's looking at a more broader level as to what happens to these communities under either a fallow, a ryegrass or a biofumigant. One of the interesting results is that we looked at what happened under the fence. So this is what actually happens on a non-cultivated site, and this is all our crop site here. So each point represents the entire community in one soil sample, and this may be 100 to 1,000 different communities. In this case, we're looking specifically at eukaryotes. So basically, this is a multi-dimensional scaling plot. The closer a point is to each other, the more closely related it is. So these populations here are distinctly different from the populations within the field and highly significant at 0.005. And that response is shown also in the bacterial and the fungal communities. Breaking that down, we can look at the proportions of various bacteria, and we can see that where we have under the fence, which is here on the right, we have a greater proportion of actinobacteria, which is this grey here. And that's to be expected because these are bacteria that decompose and utilise carbon as a source. Under the cropping part of the field, where the conditions may be more anaerobic, we have a greater proportion of bacteria, of these bacteria here. So that's kind of a DNA fingerprint of the soil at a family level. Just broadly going back, I showed earlier that we had a pH effect across the site, southern versus northern sides. If we look at the communities from these different areas, there's been a distinct difference. So the populations on the right-hand side of the line are all from the southern side of the site. The populations on the left-hand side of that line are all from the northern side of the site. So they're clustering together based on where the soil samples were collected from the field. This is looking at the eukaryote populations. Once again, it was highly significant. So it's indicating that we've got some large effects on the field, within the field, related to pH impacting the soil communities that are actually there. If we remove the pH effect, we actually see some more subtle differences. So this is just looking at the differences between our biofumigant crop, our rye crop and our fallow. And basically we have patterns where some are similar, some are not. It worked out from a statistical perspective for bacteria that there were some marginal differences, but fairly subtle differences in terms of bacterial communities when we're comparing the 3 main treatments. That's once again a DNA fingerprint of what we see from the different treatments. The essential thing to remember, differences are only subtle. There are no major changes in groups of bacteria or eukaryotes, so none of them suddenly disappear or appear. There's just relatively small changes that are occurring. Just a quick slide on weeds. So UNE came over and did some weed analysis on the site. Key point they found is that under fallow, essentially we have less weeds than under ryegrass or caliante. That was specific for chickweed. So our fallow treatment does include 2 herbicide sprays, so that's partly responsible for the reduction in weed numbers. Ryegrass was more effective at suppressing weeds than caliante. Just summarising now production over the last 4 years, and that's probably the most important thing, what actually happens in the commercial spring-summer crop. So from carrot that was grown in 2015-16, we had significant yield benefits from the grass and caliante treatment, up over 80 tonnes per hectare from the grass and caliante compared to fallow of 70.5 tonnes per hectare. You probably can't see it with how light the screen is, but there were visual differences in darkness of the plots. So there was a darker green leaf growth coming out of the grass and caliante plots. 2016-17 we grew broccoli. There was no real benefit from the grass or caliante treatment. The major outcome from this work was that we recorded club root from where caliante had been grown. That's probably to be expected. Caliante is a brassica that hosts club root, and then broccoli is once again a brassica that is prone to getting club root. So we had a greater proportion of plants from the caliante plots that had club root. So next crop was potato, 2017-2018. No difference between yields. Fallow 75 tonnes per hectare, ryegrass 72.7, biofumigant 76.5 tonnes per hectare. Just in this last year, we've had another carrot crop. Crops have been sorted into different categories, and we have done some financial or economic analysis of this. From a yield perspective, the biofumigant crop had significantly greater yields than the fallow. When we took economics into account, because of the extra costs in managing a ryegrass and biofumigant crop in terms of seed, ground preparation, termination of the crops, fallow and biofumigant ended up coupling together. Ryegrass was slightly worse from an economic perspective. There's further work to be done in this area. That's once again just a quick summary of those last 4 years. Summary of today's talk. Cover crops improve or maintain higher levels of carbon. Cover crops maintain structural stability. They prevent erosion susceptibility. Cover crops maintain increased subsequent commercial crop yields in most cases. Cover crops altered microbial community profiles. Cover crops didn't really significantly impact pathogen levels, and cover crops encouraged some weed species within this specific trial. Where to for here? We recognise that we're growing caliante as a monoculture. That's probably not ideal. Future work needs to probably use a series of different mixes. From a ryegrass perspective, that's traditionally been used in Tasmania as a cover crop, but there may be issues relating to emerging herbicide resistance in this area, and some operations are looking at replacing ryegrass with other options for grass cover crops. This work will maintain key scientific measures, specifically over the next 5 to 6 months while there's a little bit of funding coming out of the Aggregate project. But the site offers opportunities for further PhD studies and learning opportunities and collaboration with other national and international programs working on long-term soil health. That's all for today, thanks.